Structure of T4 bacteriophage. In this video, you will learn about bacteriophages or phages as they are called and their significance. You will also learn about the T bacteriophages and the structure of the T4 bacteriophage. A virus that infects bacteria is known as a bacteriophage. The word is often shortened to just phage. The term bacteriophage means bacteria eater. We know that viruses are obligate intracellular parasites as they lack the means for self-reproduction outside a host cell. Like other viruses, bacteriophages typically carry only the genetic information needed for replication of their nucleic acid and synthesis of protein coats inside the host cell. For this, they need precursor molecules, energy generation, and they need ribosomes, all of which are supplied by the bacterial host cell. Bacteriophages were discovered independently by Frederick W. Twort in Great Britain in the year 1915 and by Felix de Herelle in France in 1917. It was de Herelle who coined the term bacteriophage, meaning bacteria eater, to describe the agent's bactericidal ability. Bacteriophages also infect the archaea. T phages are a series of virulent phages that infect the bacterium E. coli. They cause a cessation, that is, they stop all bacterial metabolism on infection. E. coli is a host cell for the T-even phages, that is T2, T4, T6. The T4 type of bacteriophages, which we will be studying about in great detail, are found everywhere in nature and occupy environmental niches ranging from the mammalian gut to soil, sewage, and oceans. These are also the most well-studied members of the viral family Myoviridae, which includes the tailed viruses. What is the importance of bacteriophages? Since their discovery over a century ago, bacteriophages have been primarily exploited to control bacterial populations and to serve as tools in molecular biology. Many early discoveries in molecular genetics, such as the genetic code and the confirmation of DNA as the genetic substance, were based on the study of the T-even phages. Bacteriophage-based diagnostics and phage typing. Phage virions and the proteins they code for are useful for the detection and specific identification of bacteria, as the phage-bacteria interaction is specific. Phage display. Phage display is a technique. In this technique, a gene encoding a protein of interest is inserted into a phage code protein gene, and that causes the phage to display the protein on its outside. This technique is of great importance in immunotherapy. Due to this technique of phage display, we can use phages as vehicles or nanocarriers for delivering vaccines and chemotherapeutic drugs. Phage therapy. Phage therapy uses phages to treat bacterial infections. With antibiotic resistance becoming problematic in the late 20th century, there has been a renewed interest in phage therapy research. Let us now have a look at the structure of the T4 bacteriophage. The T4 phage is a tadpole-shaped virus about 225 nanometers in length. Based on electron microscopic studies, the structure of the T4 bacteriophage can be subdivided into the following parts: the head-tail connector or the collar or neck, tail, the base plate, and the tail fibers. So here is the head, here is the head-tail connector or the neck or collar. This is the tail, this is the base plate, and here you have the tail fibers. The head is icosahedral, which means that it has 20 faces and 30 edges. The head is made up of an outer covering called the capsid, and this capsid is made up of protein units which are called as the capsomeres. The T4 phage is said to have a capsid made of nearly 2,000 capsomeres. Within this capsid is present the genetic material. In the case of the T4 phage, this is double-stranded DNA. The DNA is quite long and is tightly packed into the head. And between the head and tail is the collar. Now this collar is a connector. It connects the head with the tail, and it also has proteins, fibers that are made of protein, and these are called as whiskers. They help to hold the tail. The tail is long and helical. It has a hollow interior, so it's hollow in the center. Here it's hollow, and it is surrounded by this contractile sheath, which is called the tail sheath. This hollow tube, that is the tail, as you can see, it has openings at both ends. And this entire tube, it can contract because of the contractile proteins in the surrounding sheath. This sheath itself is made of some 144 protein subunits. Below the tail is this base plate. The base plate is hexagonal, and the tail fits into a hole which is there in the base plate. There are 6 tail fibers or spikes that are attached to the 6 corners of the base plate. Remember, the base plate is hexagonal and the tail fibers are attached to the 6 corners. Each spike, this is a spike, this has 2 parts, the proximal and the distal. The proximal part of the spike is attached to the base plate, while the distal part of the spike, this distal part, it helps in recognizing specific receptor sites on the surface of the bacterial cell wall. The genome of the T4 bacteriophage was sequenced in the year 2003, and in the past 20 years, many T4 proteins have been studied using X-ray crystallography. Phages infect bacteria in 2 ways. These cycles undergone by viruses to replicate within bacterial cells are called lytic cycles and lysogenic cycles. In Escherichia coli, lytic infection is caused by T phages, while lysogenic infections are caused by lambda phages. We will be looking at the 2 types of cycles in detail in the next videos. Thank you.